Dilutions are prepared by adding solvents to concentrated storage or stock solutions. A simple dilution is a reduction in the concentration of a substance. We've all made simple dilutions. For example, we make orange juice by diluting a can of concentrate. This concentrate is the stock solution. According to the directions, we dilute the known volume of 1 can of stock solution with a known volume of water, in this case 3 cans, to make our final concentration. Here, our orange juice dilution factor is 4 parts final solution to 1 part stock solution, or 4 to 1, sometimes written 4x. The term dilution factor is defined as the ratio of final volume to added volume. The final volume was 4 cans. The added volume was 1 can. Serial dilutions are often useful, and as the word serial implies, they involve more than one dilution step. More specifically, the term serial refers to a series of stepwise dilutions that progressively lower the concentration of a stock solution by a known dilution factor. The dilution factor is usually constant for each step. For example, if the repeated dilution factor is 10 to 1, then serial dilutions would produce concentrations of 10 to 1, 100 to 1, 1,000 to 1, and so forth. Serial dilutions are especially useful to make standard curves to determine concentrations of unknown chemicals by spectrophotometry. Preparing a standard curve is shown in another video on this website. Let's prepare some serial dilutions. We'll use the 1 molar stock solution of cobalt chloride that we produced in a previous video on preparing 1 molar solutions. First, pipette 10 milliliters of stock cobalt chloride solution into a test tube labeled stock 1X. Label 4 other test tubes as tube A 0.1x, tube B 0.01x, tube C 0.001x, and tube D 0.0001x. Use a clean pipette to add 9 milliliters of distilled water to each of these tubes. This water is the solvent to progressively dilute the stock solution. Now use a clean pipette to transfer 1.0 milliliters from stock 1X tube to tube A. Don't submerge the tip of the pipette in tube A. Mix the contents of tube A. Tube A now contains 9 milliliters of water and 1 milliliter of 1X stock solution. The final dilution for tube A is 0.1x, producing a 0.1 molar cobalt chloride solution. Now use a clean pipette to transfer 1 milliliter of solution from tube A to tube B. Mix the contents of tube B thoroughly. Tube B now has been serially diluted by another dilution factor of 10 to 1 down to 0.01x, or 0.01 molar cobalt chloride. Now use a clean pipette to transfer 1 milliliter from tube B to tube C. Don't submerge the tip of the pipette in tube C. Mix the contents of tube C thoroughly. Tube C now has been serially diluted by another dilution factor of 10 to 1 down to 0.001x, or 0.001 molar cobalt chloride. Finally, repeat this dilution for tube D to create the final dilution of 0.0001x, or 0.0001 molar cobalt chloride. The serial dilution is now complete. The associated video on this website, titled Preparing a Standard Curve, will use these serial dilutions to prepare a standard curve that you can use to determine the concentration of an unknown solution of cobalt chloride.